All right, hey everybody, thank you so much for joining today. We're going to give some stragglers a couple more minutes to join and we'll kick things off. All right, everybody, let's get started. My name is Nick Ranelli. I'm the Regional Business Manager for Bionano Genomics here in Southern California, and we'll be moderating the session today. Thank you all for attending and being flexible with the format change. We had some rules and regulations change in light of the virus that prevented on-site gathering of groups. However, we are really excited to bring you a great presentation in webinar format. First, we'll have Dr. Sven Backlund, Senior Director of Scientific Affairs and Marketing, quickly introduce Bionano's technology for detecting structural variants down to 1% allele fraction genome-wide. Following the quick intro, the remainder of the presentation will be data from the Viore lab gathered by Ben Finlay, Director of Tumor Analysis, and Robbie Murad, Bioinformatics Scientist. They will be discussing how they use the Bionano Sapphire platform to identify novel structural variants that are relevant to the disease pathogenesis and drug resistance in patients with acute myeloid leukemia. During the presentation, if you have questions, please enter them into the question box. We'll have some time at the end for presenters to answer. Again, thank you all for joining. I'll now be passing on the presentation to Dr. Backlund to kick us off.
Hey, thank you for joining us. Nick used the word quickly at least twice, I heard, in describing what I was going to do, so I'll try to be quick. I'm going to talk about Bionano, what we do, how it works, and what we do really is called structural variants. So Bionano is not a sequencing technology, it's a genome mapping or genome imaging technology, and we call these large structural variants. We don't sequence, so we don't show you the A, G, B's, or T's. And if you're into genomics technology, you know of long-read sequencing and made pair sequencing and hide-C and all these things, but really the standard of care in the clinic to look at structural variants is this. You crack cells open and you look at the chromosomes with a microscope. And what you're really doing is you look at, you visualize patterns on intact DNA molecules, and you look at changes in these patterns to identify structural variants. And when NGS came along, there was this idea that NGS could replace all of this, but the problem is that the first step of a sequencing protocol is really this blender here. You take this beautiful intact genome and you shred it in little pieces, and so good luck telling me how many pieces of strawberry are in this blender and where they were after I turn this thing on. But that's exactly what you do when you start an NGS run, and that's why NGS has not been able to really replace these more classical methods to look at structural variants. Another issue with NGS is that the human genome is really highly repetitive, with just about a third consisting of unique sequences and 2/3 consisting of repeats. And so if you can only see a couple hundred base pairs far, you really can't distinguish between the 10,000 copies of a line, say, in the genome. And you can say, I don't care about these repeats. But the truth is that the majority of structural variants are actually flanked by repeats and are caused by the non-homologous allelic recombination of these repeats. So if you're blind to this 2/3 of the genome, you're also blind to a lot of the variants flanked by them. So that's why this is still largely the standard of care to look at structural variants. And Bionano builds on this idea. We also visualize patterns on intact DNA molecules, but we add these nanochannel arrays to this that allow for the massively parallel imaging of megabase-sized molecules. And we add labels onto these molecules, so you don't just have the couple hundred bands that you see in a karyotype, but you have 500,000 of these bands that we place all throughout the genome. And our microscope is called Sapphire, and it automates the imaging of these molecules. Bionano builds an entire workflow. We have kits for DNA isolation and labeling. We have our chips that linearize the DNA on the Sapphire instrument, and we have our own bioinformatics tools that help analyze these structural variants. So I'll tell you real quick how it works, and then I'll give you some examples of things that our customers have done with our technology. First, you need to extract megabase-sized DNA molecules, and standard DNA preps don't allow you to do that. There's just too many mechanical forces on the DNA, and it rips these molecules apart. So we developed a DNA isolation protocol that uses a paramagnetic disk that is made out of silica, just like a Kaizen column, but it has a 3D structure that allows us to hold on to micrograms of DNA, and simply just put it in a clean tube and elutes the DNA after washing. And now you have ultra pure, ultra high molecular weight DNA that's really not been exposed to any of these mechanical forces that rip molecules apart. The protocol takes just about 4 hours, so it's about the same time as a standard Kaizen protocol. We can work with fresh or frozen blood, bone marrow aspirates. We can work with about 1.5 million cultured cells, and we can work with fresh or frozen tissue as well. We're just starting early access for our new cancer protocols while working with 10 milligrams of tissue using the same fast DNA isolation protocol. And we get very, very long molecules. If you look at the slide here, you see the distribution of molecules that were imaged based on a frozen bone marrow aspirate. And you can see the N50, which is somewhat of the weighted average of the molecule length here, over 400 kb, with molecules that tail off all the way to more than 2 megabases. 2.6 is the maximum that we can image in our system. And I circled here and left where the typical read lengths are. You can see longer ones, but the typical read lengths reported in many long-read sequencing studies are just about at our y-axis there. So just orders of magnitude longer than what you can get with long-read sequencing, and because of that, we can visualize structural variants on single molecules. Once we have these megabase-sized molecules isolated, we label a single 6-base pair sequence motif all throughout the genome. We do this in a single enzymatic reaction that we're calling direct labeling, and then we stain the backbone. So again, there's no probes here. It's really just an enzymatic reaction that adds a fluorophore directly to the DNA, the same 6-base pair sequence motif all throughout the genome. And it occurs about 500,000 times in a human genome. Now we linearize the DNA molecules in our nanochannel arrays. This is really the core technology that Bionano was built on. And once these molecules are squeezed into these nanochannels, they are uniformly stretched. And because of that stretch, we can simply take a picture of these molecules and get the distance between these fluorescent labels, and that allows us to convert that then into base pairs. This is what it looks like. You can see how each of these vertical lines is a single molecule of double-stranded DNA. The backbone is stayed blue with that intercalating dye, and the green dots that you see are these label sites labeled on these molecules. And each of these molecules sit in one of these channels shown above. This is what it looks like when the molecules are pulled into our chip. You see on the left that there's Gaussian coils of DNA. Then we move that DNA through these pillar features into the channels where it gets stretched out, the molecule can relax. And then from here on, every horizontal line that you see is a single molecule of DNA that's moving into these nanochannels. I'll show this one more time from the start because it's a real pretty video. So here you see the DNA moving through these pillars to gently unwind without breaking the molecules. We use electrophoresis to do this, and the instrument actually watches this process every 5 cycles or so. And based on the salt and DNA concentration, we optimize the electrophoresis conditions to maximize throughput. Molecules are pulled in, imaged, new molecules are pulled in, imaged, and we do this over and over and over again. And that allows us to run 9 human genomes per day on an instrument for a total of 100x coverage on each of them. That is what we recommend for structural variation, calling in a typical genetic disease case a typical diploid genome. Now, if you work with a cancer sample and you expect heterogeneity, then you want to collect more data, and so you can simply run these samples longer in each of these 3 flow cells on a chip and collect 400x coverage of a human genome per 24 hours. And next week, we are releasing an update to our instrument control software that allows you to actually collect 1600x on each of these genomes. Now, this is a multi-day run, but if you need that type of coverage, you can do that for the same cost. Our cost per sample is now around $500, $450 if you run a sufficient number of them. We take the images that we collect from these molecules and we extract the digital information out of these molecules. And then on a typical sample, we perform a complete de novo assembly of the genome, which means that we align every molecule that we imaged pairwise with every molecule that we imaged. It's computationally very intense, but the result is that we built a genome structure that is really there. This is not modeled on a known reference genome. We build it completely de novo, and every single run of a human genome builds a new human genome as if we've never seen one before. It's really incredibly powerful. And then these genome maps, which really consist of the average spacing of these labels, as you see here, can be aligned to a reference genome, HG19 or 38, or a reference of your choice. And we identify structural variants just by looking at these patterns. You see on the left here that there's perfect alignment. On the right is actually also perfect alignment. Here in the middle is something going on. And if this is the reference is in green, then this would be a deletion in your blue genome map. We can call every major type of structural variants. On the left here, you see deletions and insertions, which we call starting at 500 base pairs, simply by measuring the spacing between these labels. If that spacing increases, that means some sequence was inserted there. If it decreases, that means it was deleted. We can look at repeat arrays. I'm not going to go into detail on that today, but we can measure repeat expansion disorders, for example, very easily by measuring the flanking labels. We can detect tandem duplications, direct or inverted, starting at 30 kb, by looking at patterns that occur twice in the genome. We can look at translocations, balanced or unbalanced, by building a de novo map that aligns for half with one chromosome and for half with another chromosome or another part of the same chromosome. And inversions are detected very easily. This is hard to do with other methods, but we simply see an inverted pattern often on single molecules. Once we built these maps, we visualized them in our own visualization software called Bionano Axis, and it looks like this. It's called a circus plot, and you see translocations in the middle, a copy number tool in the first circle, and then different structural variants called like that. But if you click on any of these elements, you zoom in, you can get a close-up on a translocation, for example, in this case, and we can even show you the molecules that were imaged that align to that. The copy number tool here is shown on top. But you can also export these structural variants called as a standard VCF file, and then you analyze them with any tools that you may have. I want to show you one example of a familial cancer case where 4 children in the same family had an extremely rare tumor, a 1 in a million type of occurrence. And this family has been cared for by MD Anderson and studied over a decade with every molecular method that became available, with no luck. Now, when we imaged that with Bionano, we saw something interesting in this part of the genome. We're showing you some molecules here. And you can already see with the naked eye that there's some interesting repeating pattern there. So our software built a consensus genome map that represents the average spacing of these labels. When we aligned it to HG19, we see that there's something going on there in the middle, but we also build a second map, because of course it's a diploid genome, that aligns perfectly to the reference. When you look at the controls here on top that are not affected with this disease, you see that the 2 maps align perfectly to the reference. But in these patients here, you see that something's going on, and this is actually a tandem duplication of a 38 kb fragment that's present in 6 tandem copies. Now, how do we know this so sure? We know this because we have single molecules that span the entire 230 kb of this repeat, and that is really the power of our technology. We observe these structural variants on single molecules. They're not algorithmically inferred like you would if you used short-read sequencing, say. And this gene here that we were not allowed to name is known to be affected in this tumor. So it is really an interesting find. Now, for cancer specifically, we have another pipeline. We call it a rare variant pipeline. And this allows you to do analysis on single molecules. And if you collect 400x coverage like you can in a single flow cell, we can now call these structural variants completely unbiased genome-wide at 5% allele fraction. This is for every major type of structural variants. There's no panel, there's no amplification. This is completely unbiased detection. And now with this new higher coverage that we're announcing, you can even go down to 1% allele fraction. And there's really no other technology that can do this or even comes close to this type of performance. I want to finish by giving you a few more examples from some other studies that were recently published or presented. This paper in bioRxives came out just a few weeks ago from Alexander Huygens' team at Radboud University in Nijmegen. They've always been pioneers in bringing new technologies into the clinic, and they're now doing that with us. And they ran 48 leukemia genomes of a variety of different types of leukemia and compared them against the standard of care in cytogenetics, which is karyotype, FISH, and microarray. And they found 100% concordance, meaning that all the clinically reported structural variants from karyotype and FISH and array combined in all 48 samples were detected by Bionano. This was just spectacular results there. They also looked at some complex cases like this chromothripsis case where you see that chromosome 8 really is blown up, and we can, even though it's a tough to analyze, but we map all these breakpoints and you can zoom into them one at a time if you like. They also identified 24 or 23, I think, fusion genes, of which 19 were never before reported, including one that involves BCR. Everybody knows BCR-ABL1, but here we found another translocation partner for that. So just an enormous amount of new information here. And so that brings us to this kind of idea. This slide was actually generated by one of our customers at the NHS, where you see an enormously complex workflow in a clinical cyto lab and the potential to replace all of these different methods and culturing methods and analysis tools with a single workflow, which is really quite attractive. One more case I wanted to show you here is on a hepatocellular carcinoma. This was interesting because these two tumors on the left and the right looked fairly similar with standard clinical analysis, but you see a huge difference in the genome. So if you just look at these purple lines in the middle, which are the translocations, you see a fairly normal genome on the left, and on the right, this very heavily rearranged genome. You also see in the copy number tool that there's massive amplifications of parts of the genome. And what's interesting is that we're not just showing the difference in the genomic signature of these two tumors, but we actually found the cause of that. So we detected an HBV insertion right upstream of the cycling gene, hepatitis B virus insertion. And so what happens here is that the promoter of the hepatitis virus turns on that cycling gene, forces the cells to replicate before the DNA replication is really finished, and that rips that entire genome apart. There was a recent study from UCSD on extrachromosomal circular DNA, which is fairly poorly studied, but seems to play an important role in some cancer progression. And there's more detail on this study that was presented in a poster at AGBT recently. We're putting that on our website in the next couple of days. So look at that paper or come check out our website if you want to know more about this. There's just been a beautiful use of our technology to put these structures together in these highly amplified regions of the genome. And I think that's all I have. We have a lot of videos of recorded presentations on our website, but of course we have some even better ones coming in the next few minutes. So thank you, and we can take questions maybe at the end after the other speakers are finished.
All right, now we have Ben Finley to talk about some data he was able to get at Sanford Burnham with one of our instruments. The floor is yours, Ben.
Thank you all for inviting us and for dealing with the COVID-19 induced webinar. But we're going to get to see the same data.
Sorry, guys, we just experienced some technical difficulties. We're ready to go.
It's bound to happen to me. Okay, so Sven has given a brilliant introduction to everything the system can do, but the examples I'm going to be showing today are from a project we're working on acute myeloid leukemia, or AML. So I'm not going to go over this too much, but just a little bit of background for those who aren't aware. Acute myeloid leukemia is not a very prevalent disease. There's approximately 22,000 new cases a year in the US. However, it is a major killer. Approximately half of these people will die. And just like COVID-19, this is very age dependent. If you look at the overall survival graph on the right. So if you're under 30, you've got about a 65% chance of surviving. And if you're in my age group, which I don't think is that old, it's still only a 45% chance. And this drops precipitously with age because older people have more pre-existing conditions. Approximately 30% of people will initially respond to the drug treatment, which is cytarabine and idarubicin. It's horrible traditional chemotherapeutics. But without bone marrow transplantation, almost all these patients will relapse. So there's a definite need for new drugs or repurposing of extant drugs, and also to identify target populations that may respond to these treatments. So this project is an ongoing collaboration with Scripps MD Anderson, with Dr. Jim Mason. We've been collecting samples from them for more than 5 years now. And we have approximately, I think, 76 samples. So we receive them, we isolate the PBMCs, and we do our chemical sensitivity analysis, where we treat them with all the FDA-approved oncology drugs, or almost all, and then experimental drugs or other drugs that we are interested in. Then we send that data back to them, and they do whatever they want with it. But this is a powerful data set, so we're also acquiring traditional RNA-seq, and in collaboration with Bionano Genomics, we've run, at this stage, only 8 of our samples through the Sapphire system to get structural variants, but we intend to run much more. And our bioinformatics team, Rabi, who will speak after me, are going to show how they've integrated these findings through the chemical data. It's just an overview of the first 6 samples that were run by Bionano Genomics. And I think on the top line, you can just see the amount of data that gets collected, of the order of 1.3 trillion base pairs. And the average read length is in excess of 300 kilobase pairs. So these are very good data we can achieve. When we gave them 6 samples, we gave them 3 that we knew had major rearrangements. So a deletion of chromosome 7 in the first one, a traditional BCR-ABL Philadelphia chromosome, and then also a rearrangement in the CBFB gene rearrangement deletion. And I'm going to show examples of all of those. And yes, and the Sapphire system found all of these, of course, but it also found a lot more. If we look in the bottom, there's at least 60 or 70 different insertions, deletions, duplications in all of these samples. And these have all been filtered for strong molecule support and novelty to the healthy sample database. So these are probably not somatic. These are probably exactly due to the tumors themselves, the cancer itself. So once you've run your sample through the Sapphire machine, you just hit show me the variants, and you get thrown to a screen like this. This is from the Bionano Access software. It's very easy to use. Even I can use it. And it gives you these publication quality figures right away. So I've also added in the known canonical genes from the latest build of the Human Genome Project in blue here. And I further filtered these to only show structural variants that have overlapped with known canonical genes. And we're showing this example here because it has this translocation BCR-ABL Philadelphia chromosome, but another interesting feature here is a deletion of the Y chromosome in this sample, which I've never really heard of before, but maybe of interest. And as Sven showed earlier, once I'll go back. Any of these here at all, the insertions, deletions, inversions, translocations, you can put your mouse over, hit click, and you go to the map. Again, as Sven showed, the actual build made by Sapphire is in blue, and the reference sequences are in green. We can see this here tracking along chromosome 9 through the ABL gene and then stopping and then remapping to BCR. Then on the bottom, we can see the actual individual reads. What we see here in blue are reads that cover this exact region. So that this translocation seems to be prevalent in almost all the cells in this sample. Another example from the same sample, sample 13, is showing a deletion. Again, it's showing blue is the reference sequence, or blue is the map made by Sapphire, and green is the reference sequence. Here we can see a deletion of a ring finger gene, and also this other gene, EBF1, which is known to be involved in differentiation of hematopoietic cells, so could conceivably be involved in tumor development. And up at the top, we see a computed algorithm of copy number variation, which again agrees with the map made that there is less, and there's deletion of this gene. This is just another sample. This is number 27. Again, we knew this sample had a fusion in chromosome 16, and Sapphire detected that. It also detected another translocation down in chromosome 8, and again, it detected lots of inversions, deletions, translocations. And this is just this zoomed-in map of that translocation in chromosome 16 on both sides, on the P arm and the Q arm, because it goes around the centromere. We see disruption of this MTH11 gene, which codes for myosin. It's a deletion and an inversion. And then we can see on the other arm of the chromosome, we again see this deletion on CTCF, which is a negative regulator of c-Myc, and also disruption of the CDFB gene, which is a master regulator of hematopoietic genes. And again, these are supported by the copy number calculations, where we again see this deletion of one copy of these genes. Another example is this sample 9. This sample is actually from a relapsed patient when we got it. So the original cytogenetics had detected chromosome 7, which again, Sapphire detects. However, it also detected 2 other structural variants. Now, these might have been missed by the original cytogenetics, or these may have been a low-frequency clone that has actually come back up in this patient when they relapsed. So this shows there's a potential for disease progression mapping or clonal evolution analysis that can also be done. And of course, Sapphire detected, again, lots and lots of insertions, deletions, and so forth. And this here is just, again, the zoomed-in version of what this translocation is. It's in the imaginatively titled KIAA1377 gene. And you can see it's tracking along chromosome 11, deletion, and then tracks chromosome 2. And the bottom panel is just the same from the opposite direction. And this gene encodes a protein called CEP186, which is involved in microtubule organization, which again could be very relevant for the disease. Another example I wanted to show is this one again from sample 9, where we see a deletion in FOXP1 gene and probably a disruption of this elongation initiation factor. But what's interesting about this one, not just that it's novel to the Bionano sample database, if we look at the copy number, it's giving us a copy number of about 1.65. And if we look at the actual reads from the map below, in blue are the actual reads that directly cover this region showing the deletion. They're probably around maybe 15% of the reads, whereas the other reads are not mapping over this entire region. So this suggests there's an actual clonal population in here, and that approximately 15 to 20% of this sample has this deletion. So it again shows the sort of power of this technology, as Sven mentioned earlier. I don't think there's any other way one could find this information. I think this is my last example. We also had spiked in samples that had no traditional large-scale cytogenetic abnormalities. This is sample 26. And of course, Sapphire doesn't find any either, but it does find a lot of deletions. This sample seems to be very deletion-rich, but there's also a lot of insertions. If we just click on any one that we may be interested in, I chose this one, in lysine methyltransferase 2A, and what we can see is we can detect, with a level of granularity too that is not possible with our technologies, we can get an almost 32 kilobase pair insertion into this lysine methyltransferase. And this is a gene that is well known to be involved in some AML. So that was just a very sort of rough overview of some of the examples. I think I showed 1 or 2 from each sample, but as we saw in one of the initial slides, there's much more data there. So rather than picking through all our individual samples, we thought we will hand this over to our bioinformatics specialists. So in summary, we found again 100% concordance with previously detected cytogenetic events. This technology is certainly the potential for gold standard to overtake traditional cytogenetics, but we detected lots more structural variations. Now the key is sort of what is the significance of this, and can we associate this with the chemical sensitivities or resistance we see? I'd like to thank everyone from the Vorga Lab and Shared Resources. Of course, this is a collaboration with Scripps MD Anderson, and also all the team at Bionano Genomics. And with that, I'm going to hand over to Rabi Murad from SVP Bioinformatics, who will show how he is integrating our chemical data with the structural variants detected by Sapphire.
Good afternoon, everyone. Thank you for attending this webinar. My name is Rabi Murad, and I'm from the SVP Bioinformatics group. So I will talk a little bit about the approach that we want to take with integrating the different multi-omics data sets that we have for the adult AML patients cohort that Ben talked about. Over here, I'm just showing an example from the pediatric AML cohort that was published in 2018. Although we know that the pediatric AML patients, the frequency of the mutations are different from the adult cohort. The point here that we want to make is that the structural variants play an important role in the etiology and the pathology of AML patients. So as you can see on the frequency of the different types of molecular aberrations that were observed in the pediatric AML patients, there are many examples of structural variants. And on the right, you can see that the molecular aberrations related to the structural variants explain the survival rate of the AML patients in pediatric cohort. So there are certain structural variants that confer better outcome for the AML patients than others. So the point to make is that structural variants play an important role in AML pathology. In our case, as Ben mentioned earlier, we have different types of data sets that we would like to eventually integrate in order to inform drug sensitivity in the AML patients based on the types of structural variants present in these patients, and also on the gene expression profiles of these patients. So this is very preliminary results since we don't have statistical power yet based on the number of patients that have been assayed for structural variants, drug sensitivity, and gene expression. But as Ben mentioned earlier, for the structural variants, we have so far assayed 8 patients for structural variants using the Bionano mapping technology. For the drug sensitivity, we have a almost complete data set. And for gene expression, which is currently in the pipeline, that are being sequenced. But so far, we have data from 16 patients that's coming up. And we're also thinking about looking into coding variants by exome sequencing, but that's something that we are actually looking at right now. So Sven gave you a very good explanation of the Bionano mapping technology earlier, so I would skip this. But the basic idea is that it gives you a very reliable assay of the different types of structural variants present in the patients. In this case, we are looking at the AML structural variants in our AML cohort. So this is a summary of the 8 patients that have been assayed for structural variants using Bionano mapping. And as you can see that for most of these patients, through karyotyping and genotyping, we know we have some information about major structural rearrangements in these patients. So this is an example of one of the structural variants that was detected in one of the patients that we knew through karyotyping that had BCR-ABL fusion. And you can see that this example shown earlier by Ben is also was detected by Bionano mapping technology. And this is a summary of all the different types of structural variants in our 8 patients, AML patients, that were detected by Bionano mapping. And I should mention that these are called the rare structural variants. So these are the type of structural variants that were not observed in normal human population. That's the database that Bionano has on hand. And I should also mention that the type of structural variants that were detected by Bionano have complete concordance with the structural variants that were detected through genotyping and karyotyping in these 8 AML samples. So as I mentioned earlier, the one type of data set that we have for these patients is drug screening. So these AML patients were screened for sensitivity to small molecules libraries So these are the three types of libraries that we have: the oncology drug libraries, the ODL, ODL2, and ODL3. So different patients were screened using one of these screening libraries. But the point to make here is that for most of these small molecules, there's overlap for all three libraries. So 94 small molecules are shared between, and we are planning to use those 94 molecules for downstream analysis. And then this is a summary of the types of targets of these small molecules in the ODL libraries. So the biggest class of the drugs, these are related to DNA damage, and you can see that there are also targeted small molecules that have targeted targets in the oncology. So out of the 8 samples assayed by Bionano, those samples are also screened for these ODL libraries, and I'll show you some of the results from that, preliminary results. And also some of these samples were also screened for the epigenetic library. So there are 335 drugs in this library, targeting mostly epigenetic markers. And for the analysis of the drug screening data, we are using the measure called the area under the curve. So one of the common measures of drug sensitivity is the IC50. It's the concentration of the drug that shows a 50% decrease in viability of cells. But in our case, we have 3 data points, and AUC is a more robust measure of drug sensitivity, and that's what we are using for our analysis. So this is the result of the drug screening of the ODL libraries for the 8 patients. As you can see to the right of the heat map, so this is showing the AUC for these small molecules for the 8 patients. And you can see that in the red color, that's an AUC more towards the 0. That shows those represent the small molecules that are showing decrease in viability of these cells. And you can see that there's a big cohort of small molecules to the right, mostly related to DNA damage, that show that most of these samples show good sensitivity towards. But another point to make over here is that for these samples, for some of them, we have the patient before and after the complete responder and the non-responder versions of these patients, the relapsed versions. And you can see for 2 of the examples that I'm pointing over here, the patient number 42 and 66, the responder and the non-responder versions, they cluster together. But then there are examples. There's one example where the non-responder, the drug sensitivity is different from the responder version. And over here, I'm highlighting some of the drugs that are sensitive, that the non-responder is sensitive to. And hopefully this type of analysis will inform the drug sensitivity of such patients. And we also noted that for some of these patients, we have the corresponding drug sensitivity data from the blood and marrow samples. And you can see that they usually are very similar, so there are no difference. And so in this slide, I'm just showing some of the genes that were affected by the structural variants that were mapped through Bionano mapping. And in this case, since our sample size is still smaller, and we're only showing the genes that were affected by structural variants in at least 2 samples. And over here, I'll highlight the example that we had. So for 2 of the patients, we have BCR-ABL fusions that are being pointed over here. But as you can see, the overall types of genes that are affected by structural variants are very different from these patients. Although they carry BCR-ABL fusions, the rest of the genes that are affected by these structural variants are not shared between these 2 samples. And this is also reflected in the sensitivity of, drug sensitivity of these two patients. And over here, I'm highlighting those two patients that carry BCR-ABL fusions. You can see that their drug sensitivity, based on drug sensitivity, they no longer cluster together. So this shows that the types of genes that are affected by structural variants and that lead to differential drug sensitivity can be well informed by mapping the, giving a comprehensive mapping of the structural variants in these samples. And also, so since by now, as I mentioned, that we have, we're looking to expand the real structure variant and gene expression profiling of the adult AML cohort to increase our statistical power and eventually integrate the multi-omics, the RNA sequencing and structure variants that are in the pipeline in the future, and integrate them with the drug screening results to provide a functional interpretation of the drug sensitivity results. So for the future multi-omics data integration, we're planning to use machine learning algorithms such as Diablo. So as you can see on the left side, in case of a single data set, one of the most commonly used algorithms that can be used as, for example, principal component analysis. But disparate data sets from the same patients, for example, gene expression, drug sensitivity, mutation analysis, these can be integrated in a similar fashion as a principal component analysis in a single data set. The idea would be to look at the clustering of these samples based on the different types of profilings of, for example, mutation, drug sensitivity, gene expression, and so on, and look at each component of each data type that leads to such clustering of these data sets and inform the sensitivity of these cancer patients to different drug types. So I'd like to acknowledge Ben Fenley from Viori Lab at SPP and all the folks at the Bionano Genomics who have had great help in establishing the structural variants mapping in these AML cohorts.
All right, thank you, Sven, Robbie, Ben, for presenting on the technology and its capabilities and giving everyone a great understanding of the data and how it's being used in the field. So far, we don't have any questions, but I want to give all the attendees an option to write some questions for our presenters in the questions box for them to answer while we have some time. So we have one question here in the box, and this could be answered by, I believe, Sven is probably the best one. How high is the cost per sequencing sample?
Yeah, so it depends a little bit on the path that you want to use to run samples. So we provide a service in-house where we run cancer samples for $750. If you own an instrument, then it's as low as $450 per sample. And if you want to have an instrument placed in your lab as a reagent rental model, we're charging $550 per sample with a commitment of 120 genomes minimum to get started. Now, of course, if you are at Sanford, Burnham, maybe you can go talk to the Viori lab and make a deal with them. So maybe they can help you out. I get a thumbs up here.
Yes, if you're at SPP, as part of, we actually own the Sapphire system now. It's part of our shared resources. And as part of our acquisition, we acquired enough reagents for 60 genomes. So there's a potential there for almost cost-free analysis. And we're happy to talk with any SPP people about that.
So next question in the box here, could this technology be applied to microbial DNA?
So yeah, that's a good question. So in theory, yes. in reality, I think there are some limitations on the value that we provide. You know, our strength is megabase-sized molecules, and sure, that works for bacterial genomes, but you can realistically do bacteria and metagenomics with PacBio as well. We have, as far as I know, one published paper fairly recently on a metagenomics project using bacteria where different antibiotic resistance causing mutations were mapped with Bionano on a population sample of bacteria. So it's definitely possible. Talk to us if you have a specific project in mind, and we can maybe sit down and figure out whether it makes sense or not.
So next question coming from Peter Adams at SBP. Very interesting data. I assume MDM2 inhibitors are in your drug collection. Does your study tell you anything about sensitivity of AML to MDM2?
Hi, Peter. Yes. So although the data Ravi showed today was mostly the oncology dose library, we have also used the epigenetics collection that we've been sharing with you guys. So that data is in there, and we're happy to share and happy to collaborate as usual.
And one last one from Peter. How are you distributing the free reagents? First come, first served, internal review? Have you guys had that discussion?
We have not had that discussion, but happy to meet to discuss. But you'd definitely be welcome to be one of those people.
Awesome. One from Robert Weschler-Rea. You mentioned Paul Michel's ecDNA study. Are you able to detect extrachromosomal circles and distinguish them from intrachromosomal amplifications?
Yeah, so what's actually interesting about their study is that they developed their own software to do exactly that. So they wrote a software called Amplicon Reconstructor, and it actually combines Bionano data with Illumina data that they have on these samples to really stitch together all of these breakpoints and then fill in some of the shorter fragments based on the Illumina data. And with that software tool, they're perfectly able to circularize that circular DNA and distinguish it, I think, from the amplifications that they see within the chromosomes themselves. So it's really their software that enabled that, but it's pretty impressive.
One more question. Apart from the data from the Hoyschen paper, are there any other data comparing Bionano data with standard sequencing techniques?
So there's a number of papers from consortia, including the Human Genome SV Consortium, that compared our sensitivities for structural variants against PacBio and Illumina mostly. And so what they found, I have some slides on that, but I don't think I have them handy right here. We can send it to you later. But what they found in that paper is that really insertions in general are extremely hard to detect with sequencing technologies, even with PacBio. And so we really cornered that. Everything above 1 kb that's an insertion is almost exclusively detected by Bionano. Deletions are often a little easier to see with sequencing, but even there, a large fraction of larger deletions are missed entirely. And that is often because the flanking sequences around these deletions are repeats. And so, again, our strength is these megabase-sized molecules that can identify any type of structural variant, even when they're flanked by large repeat arrays. So there's a couple of those papers, and I think we'd be happy to send them to you afterwards.
All right, any last questions from the attendees? Awesome. Well, if you'd like to connect with the Viore Lab at SBP or Sven and ask additional questions, are interested in learning more about the technology, or want to pursue a project of your own, please reach out to me directly. My contact information will be in the follow-up thank you email, as well as a copy of the webinar presentation for your own review. We have included our Sapphire product brochure and 2020 product offerings in the handout sections of the webinar app. Please feel free to download these, as they contain a lot of good information if you want to pursue your own project. With that, have a great rest of your day, and we'll be officially concluding this webinar. Thank you guys all, and appreciate the flexibility in us changing the format again.